Swayam Prabha. Digital India. Educated India.
So, hello everyone, today I will start module 6, that is cell surface interactions. So, in this module we will see host response to biomaterials, then cell adhesion, and followed by how we define the biocompatibility of a biomaterials. So, first we will recap the last module. In the last module we discussed about the aggregation of proteins. So, how protein aggregates in the solution, also in the presence of a surface, and what are its effects. So, if there is a aggregation of proteins, so it start with the conformational changes. So, the protein become functionally inactive, and also it is associated with various neurodegenerative diseases like Alzheimer, Parkinson and prion. Then we discuss how we can characterize the protein aggregation in terms of fluorescence intensity, then residual protein concentration, then hydrodynamic size of the aggregate, morphology of the aggregates. Then we quickly discuss what are different factors affecting the aggregation of a protein in the bulk, also in the presence of surface. So, in the bulk we discuss effect of pH, temperature, ionic strength and some denaturating salts. In the presence of surface we discuss the effect of shear and effect of adsorption. Then we discuss what are the different short range, long range and electrical double layer forces responsible for the aggregation of a protein. Then after that we discuss what are different pathways, how protein molecule converts from a native state to aggregated state. So, we discuss three pathways, from native to intermediate to unfold to the aggregate, then from native to directly to the aggregate, and one was from the native to its degraded component, then to the aggregate. Then we discuss how we can analyze the kinetics of aggregation in the bulk. We wrote the kinetic equation and how to solve it to calculate the rate of aggregation, also how to determine the value of lag time and growth rate. Also we discuss from the experimental results, particularly from the TST fluorescence intensity as the residual protein concentration, we can determine the rate of aggregation simply by fitting the sigmoidal curve or an exponential expression or a linear tangent in the growth phase. After that we discuss particularly how the presence of surface affect the aggregation of a protein. So, there we discuss different surfaces formed by the self assembled monolayers, and we choose a model protein insulin. So, in the presence of surface we saw how its adsorption varies. Then from conformational changes to formation of multilayer adsorption or nuclei, then to the formation of the aggregates. So, there we discuss in terms of TST fluorescence intensity, we discuss in terms of adsorption, we discuss change in the secondary structure. So, in the secondary structure we observe the insulin was having a about 84% of alpha helix, means alpha helix rich protein, and after aggregation the alpha helix was completely absent and it was mainly converted to the beta sheet. Then we also saw the morphology of the aggregate, and we found that the surface with more affinity of the protein, it means higher adsorb mass, resulted in faster aggregation. It means it has the least value of lag time. So, in today's lecture we will start with cell surface interactions. In the cell surface interaction, particularly for the biomaterial, when we implant the biomaterial, then it comes into contact of body fluid. Then for the intended function of a particular biomaterial, it is desirable to have the cell surface interaction. So, we will just see what are different steps involved in the cell surface interactions, and during the implantation or the cell surface interactions, or you can say the application of a biomaterial, what are the different steps involved, particularly related to the host cell response. Then what are the sequences in the host cell response, what are the different pathways for the inflammation, and we will briefly discuss what are different factors which affect the cell surface interactions. So, as I told, whenever a biomaterial comes into contact of body fluid, then the surface of the biomaterial interact with the body fluid. So, body fluid we can say it is composed of water molecule, protein molecule and cells. So, depending on their diffusivity, very first step is the surface water interactions, then surface protein interaction, and followed by the cell surface interaction. So, as it comes 1 to as a third step, so sometime this cell surface interaction it looks like the protein and cell interactions, because already adsorbed protein is present on the surface. So, these are the 3 steps, and all these 3 steps are regulated by the, or you can say they are all dependent on the surface property, including the its mechanical, chemical and surface property of a particular biomaterial surface. And of course, depending on the each step, means the surface water interaction will affect the surface protein interaction, and surface protein interaction will affect the cell surface interaction. So, all these 3 steps are also interconnected. So, you can see this is quite a complex network of different processes, and ultimately the intended aim of a particular biomaterial implant is to perform its desired functions without generating the host response. It means it should not behave like a foreign material. So, when a cell comes into the contact of the surface, it contacts take place between the outer layer of the surface and outer layer of cell is we can say cell membrane. So, that interaction between cell membrane and outer layer of cell, it decides the nature of adsorption of the cell and the surface, whether the cells are going to secrete some particular proteins, particularly the extracellular matrix. It also decides whether how transportation of different molecules, and again the communication between the cell membrane and the internal organs of the cell. So, particularly the cell adhesion is communicated by the signal proteins. like integrins. So, you can say the outer layer of the surface and outer layer of the cell membrane interacts, and that interaction decides what is the type of interaction, and once the cell adhesion takes place, then after the attachment and the surface attachment, we can say this is like a adsorption or adhesion, and after adhesion the cell molecules grows, it proliferate, or you can say this is the spreading of the cell on the surface, and followed by that it goes for its differentiation. So, this goes with time, and it take different time intervals starting from days to weeks. So, now we can have two types of biomaterials, one is degradable, other is non-degradable. So, when we implant a biomaterial, so for that it has to go through a surgery route. So, it causes the tissue damage, and when there is a tissue damage, so it activates the immune system, and also as I told, there will be the Raman effect because of the diffusivity of different body component. As I told, there will be the surface protein interaction followed by the cell surface interactions, and depending on the surface protein interactions, it will govern the cell surface interaction, particularly if during the adsorption, if the native conformation of that protein is lost, it means the protein is denatured on the surface. So, that is not going to facilitate the desired cell surface interactions, and sometimes it leads to the contamination leading to biofouling, and finally, the rejection of the implant. So, this protein surface interaction and cell adhesion, it leads to the formation of temporary matrix formation, or you can say the provisional matrix formation, and these two finally cause the immune response. You can say the innate immune response due to the injury and due to the presence of biomaterials, and this goes with time. Again, this time vary from the few minutes to few days, it continues till the weeks time. And finally, what is the possible outcome? If it is a non degradable material, it means it will be present inside the body for a longer duration. So, it causes the tissue integration, the foreign body interactions or foreign body reactions, and encapsulation. But if the biomaterial is degraded with time, so it causes the constructive remodeling. Of course, the foreign body reactions will be there whenever any foreign material coming inside the body, and also it causes the fibrous scar. So, these are the possible outcome in case of non-degradable and degradable material, and whenever the foreign materials or foreign body comes, so it causes the inflammation, and inflammation serves to contain, to neutralize, to dilute or well off the injurious agent or that particular process which is causing the inflammation. And how it happens? The inflammation mechanism goes through the signals, or you can say it is a cascade of different steps which causes the inflammations. So, as I told, there will be the host cell response whenever any foreign materials, foreign body, whenever any foreign body is implanted, whenever a foreign body is implanted, then we go with the host cell response, and this host cell response having the two part, one part is due to the presence of material and other is due to the tissue injuring during the implantation. So, this tissue injury during the implantation, it is simple like a wound healing process, and so you can say this is a quick process. It is like you got a injury and injury is healed with time within a few days to week, but in the presence of material it depends whether it is a degradable material or non-degradable material, but this response is a persistent response, because this material stay for a longer duration inside the body, and as long as the material is present, that foreign body or host cell response will be present. So, this is a persistent inflammatory response. So, this persistent inflammatory response, it depends on what is the life of that particular biomaterial. Now, whenever there is a host cell response, so the body is having a mechanism called homeostasis. So, due to this homeostatic mechanism, there is some sort of disturbances because of this implantation and surgery. So, this homeostatic mechanism are disturbed or perturbed, and the pathophysiological conditions are created, and those conditions are resolved by the host reaction of a biomaterial, and how much it is resolved, or you can say how effective is the homeostatic mechanism, or how efficient is the homeostatic mechanism after the implantation, it ultimately determines the biocompatibility of a particular biomaterial. So, we can say whenever there is injury or implantation, so the homeostatic mechanism of body is disturbed and pathophysiological conditions are created, and these conditions are resolved by the host reactions to a biomaterials, and depending on the balance between how much it is disturbed or perturbed and what is this pathophysiological conditions, ultimately it decides whether your material is biocompatible or not. So, we will see what happens during the homeostasis. The first thing is that vascular spasm, this is the first step. It means the blood vessels are damaged during the surgery, then this lead to the formation of the platelet plug, and followed by the third step is the activation of the coagulation cascade, or you can say the clotting factors. So, particularly like you are having a fibrinogen, and once the thrombin is activated, it will convert to the fibrin, and now fibrin is insoluble, it lead to the formation of clot, or you can say the clotting. And then fourth step is once fibrin is formed, the formation of the fibrin plug or the final clot. So, these are the steps involved during the homeostasis, and body is having a process called fibrinolysis, in which the clot is degraded in a healing vessel. So, body is having this mechanism, there will be formation of the fibrin plugs, and the fibrin plugs are degraded by the mechanism or the process we call the fibrinolysis. Now, apart from that, there will be activation of complement pathways, and so we will quickly discuss about what is the complement pathways. So, these are activated through a triggered enzyme cascade, or you can say different enzymes will be involved during the activation of these complement pathways. So, we see there are three pathways: classical pathway, lectin pathway and the alternative pathway. So, in case of classical pathway, it is initiated because of antigen antibody immunocomplexes. This lectin pathway is activated whenever there is a presence of particularly the carbohydrate binding domains of that particular pathogens are available. And third pathway is the alternative pathways. It is activated when there is a some pathogenic surface or even some foreign materials or some spontaneous hydrolysis. So, particularly when we go for this foreign material, in case of foreign materials, this alternative complement pathway is activated. So, we will see how does it work, because it is not automatically activated whenever there is some foreign material or some external stimuli. So, in case of classical pathways, the first it is initiated by the C1q, it binds to the antibody, then it activates to the C11R and C11S, and then C11R and C11S, it cleaves the C4 and C2, and finally it results into the C3 convertases, and C3 convertases have two, C3A and C3B convertases, and C3B convertases also lead to the formation of C5 convertases. So, this is the classical pathways. Now, in case of lectin pathways, there is a mannose binding lectin MBL. It interacts with the that carbohydrate motifs of the particular pathogens, then it activates the MBL associated serine proteases, and this particular proteases again it cleaves to that C4 and C2, it leads to the formation of finally C3 and C5 convertases. Now, in case of alternative pathways, that C3 is automatically hydrolyzed or quickly hydrolyzed to the C3H2O. Then here it interacts with the factor D, factor B or microbe surface, and because of this interaction the C3 hydrolyzed C3 is converted to C3B, and again in the presence of particularly this factors B or D, or you can also the properdin, the C3B is converted to the C3 and C5 convertases, and in all three pathways you say they end up with three final responses or three final outcomes, that is the inflammation. So, for the inflammation C4A, C3A and C5A are responsible. Lysis, or you can say the killing by the rupture of membrane. So, from C5B to 9 are responsible for the lysis, and this is opsonization. For opsonization C3B is responsible. So, in all three pathways it is having a initiation, then amplification, then termination, and they all end up with the common three steps: inflammation, lysis and opsonization. So, what we are seeing is this pathway is having C1q, lectin pathway is having MBL, mannose binding lectin, alternative pathway is having this hydrolyzed C3 molecule. So, these proteases are activated whenever there is a external stimuli, and finally they are activated by the proteolytic cleavage, like C4 and C2 are the proteolytically cleaved to get the C3 and C5 convertases. So, these proteases which are there in the inactive form, we also call them as a zymogens. So, they are already present inside the body, but they get activated only after the proteolytic cleavages, and that proteolytic cleavages is activated because of this antigen antibody immune complexes or some recognition by lectins or in the presence of pathogenic surfaces, foreign materials or spontaneous hydrolysis, and ultimately these all three component pathways end up with inflammation, lysis and opsonization, and all the complement pathways converge at the membrane attack complex. So, ultimately what is happening here, because we are having some foreign material which is coming inside the body, so it gives some foreign body response, and for foreign body response these are the complement pathways, the classical, lectin and the alternate pathways. So, when we go for this implantation, so these are the series of event, or you can say these are the sequences. The first sequence is injury, this is followed by the acute inflammation, then chronic inflammation, followed by the granular tissue, then host body response, and then finally fibrosis. So, if you just see if there is a injury, maybe very in the minute, then acute and inflammation and chronic inflammation they go up to, let us say, up to 72 hours, and this host body response or fibrosis it goes up to weeks. It depends, these are the persistent and what is the life time of that particular biomaterial, or you can say that particular foreign body. So, during the injury what happens, the vascularized connective tissue are damaged. So, whenever there is a damage, then the blood proteins they flow towards the damaged site, or you can say that is a process of exudation. In case of acute inflammation, again the exudation of the fluid and plasma proteins and this immigration of the leukocytes, this causes the acute inflammation. During the chronic inflammation, the presence of macrophages, monocytes and lymphocytes are involved during this chronic inflammation. Then followed by the chronic inflammation, there is a granulation tissue. It means there is a proliferation of the fibroblasts and the vascular endothelial cells, they lead to the, they end up with the granulation tissue, and then host body response, this consists of macrophages, fibroblast and capillaries, and finally in the last step of fibrosis you can say this is a fibrous encapsulation are replacement by parenchymal cells or connective tissue. So, it depends what is the intended application of the biomaterial. So, it may be finally replaced by the parenchymal cells or it may be replaced by the connective tissues. So, now all these steps are, you can say this is a sequence of foreign body responses. So, these causes the inflammation and these inflammations are mediated by the proteases, cytokines, free radicals and various growth factors. Now, when we go for particularly biomaterial implantation, so during the injury and the acute inflammations, two steps are also involved. So, that first one is in between these two we are having first blood and material interaction, and followed by the blood material interactions we also have the matrix formation. So, particularly after the injury means when the biomaterial is implanted, these are the two steps also in between the blood material interactions and this provisional matrix formation comes in between the injury and the acute inflammation. And these, as I told, these inflammations are mediated by the different proteins or proteases, cytokines, also the free radicals and growth factors. So, here I have presented the timeline. That timelines, if we just see, it starts let us say from the minute it starts with the acute inflammation, then it will go to the chronic, from the acute it will go to the chronic inflammation, then it will go to the granulation tissue, then finally last step will be the fibrous capsules. So, here you just see the intensity. So, acute inflammation, its intensity start in the very few minutes and slowly this inflammation is decreased. Now, during the chronic inflammation again we will having this leukocytes, it will be there, again it is going to decrease with time. Now, what is happening here is the, we just see the macrophages, you just see this neovascularization or foreign body gained cells. So, particularly this macrophages and this FBGC, this foreign body gained cells FBGC, they are the part of foreign body reaction. And the final step is the formation of fibroblast and this fibrosis. So, you just see it start in the minute, then it will go to the few days, you can say 2 to 3 days, then it goes to the weeks, and here the weeks time is, it depends what is the life of that particular biomaterial. So, now ultimately what happens, whenever a biomaterial implant is inserted inside a body, the first step is the, you have to go through some surgery. So, it is like for body it is like injury, then followed by that material. So, there are two responses, both one response is coming from the injury, other is coming from the material itself. Now, depending on this the cell surface interaction, there is a host body response, or you can say the host response, that is actuated by the cascade pathways and also by the complement pathways. So, in the complement pathways, we are having this classical pathway, lectin pathway and alternative pathway. So, these are the interaction or host response which is coming from the particularly from the cell surface interactions. Now, if as we know the cell surface cell material interaction is dependent on the surface protein interaction. So, if the surface protein interaction is not desirable, as I told, it may also cause the biofouling. So, for that there will be some extra responses, or it will also contribute to the host responses or foreign body responses, and apart from these two, they are also susceptible to the contamination. So, there may be some growth of pathogens, growth of some, you can say, the bacterial contaminants. So, that also causes the activation of complement pathways, or you can say it also contribute to the biofouling and you can say the host response. Now, what are the different factors which affects the cell surface interaction? The first thing comes with the what is its composition of the biomaterial, whether it is toxic or it is with time it is releasing some toxic components, what is its mechanical property, whether it is similar to that particular body part where that biomaterial implant is placed, is there any mechanical incompatibility or not. Then what is its surface texture, what is surface area, what is surface porosity, because these directly decides the contact area with proteins as well as the contact area with the cells. Now, the surface ligand landscape, how the surface ligand binding is taking place, and then whether your biomaterial is degradable or not. So, depending on, as I told, for the degradable material we ultimately final outcome is different, for non degradable material the final output is different. And finally, it is important that that material should resist the infection. It means whether it is susceptible to the bacterial contamination or not, and this is obvious that during the implantation whether it is properly placed or not, that also causes the host response, or you can say that foreign body response, and which may lead to that replacement or the failure of that particular implant. So, these are the key factors particularly for the biomaterial implant which regulates the cell surface interactions. So, I will conclude here this lecture. So, tomorrow we will continue specifically on the cell adhesion. So, today we discussed whenever there is a adhesion of cell, or you can say interaction of cell with the biomaterials, it comes into the three steps: water surface interaction, protein surface interaction and the cell surface interactions. And after the cell surface interaction, the first step we call the attachment or adhesion, followed by its spreading, growth, proliferation and the differentiations. And parallel to this implantation, there is a host cell response. It originates from the surgery or the injury, also it comes from the presence of material. So, during this foreign body response, the first step is the injury, then blood material interaction, then provisional matrix formation, followed by the acute inflammation, then chronic inflammation, then granulation of tissue, or and after the granulation there is a, you can say, foreign body reaction, and finally the fibrosis. So, these are the steps, and once the inflammation, once there is some foreign body, the inflammation is started to self regulate that particular reaction by the body. So, these are inflammations is coming from the cascades of steps or cascades of enzymes are involved in that inflammation. And then also we quickly discuss about what are the complement pathways which activate the inflammation or lysis or the opsonization. Thank you.